name is Dr. Paul Corley, and I am quite happy and honored to speak with you today. My subject is the regulation of HIV gene expression by glucocorticoid hormones and interleukin-1. Before I begin, however, I'd like to give you a little background on my perspective and approach to this most interesting and complicated subject. I am a clinical assistant professor of psychiatry at the State University of New York Health Science Center at Brooklyn, and I work at the East New York Diagnostic and Treatment Center, which serves a patient population in the East New York section of Brooklyn and the surrounding areas. This is a socioeconomically disadvantaged community that has been absolutely devastated by HIV infection and AIDS. East New York is primarily an African American and Latino neighborhood that has one of the highest rates of HIV infection in New York City, and is overwhelmingly transmitted there by intravenous drug abuse and heterosexual contact. As a psychiatrist working closely with this patient population, I am part of a team providing interdisciplinary medical and psychosocial services. I believe that I have an unique perspective and have the freedom to view the process of HIV infection in a well-rounded manner, incorporating clinical observations with virology, cell biology, endocrinology, and genetics. I find it most interesting that today's presentation is totally based on documented observations that both individually and collectively support the theory that HIV pathogenesis is mediated by glucocorticoid hormone synthesis and by various associated cytokines, particularly interleukin-1. This is a clinically testable hypothesis that may lead to an effective, relatively inexpensive treatment for AIDS that may be of benefit to everyone, including the economically disadvantaged, as well as to those infected in the developing world. Finally, I would be the first to admit that the theory is somewhat unusual and certainly has not been welcomed with open arms by the scientific community in general. Yet, it seems to fit like a hand in a glove, in my opinion, though I will let you be the judge of that. After 16 years of the acquired immunodeficiency syndrome epidemic, or AIDS epidemic, research and therapeutic efforts have primarily emphasized the role of cell surface antigens in AIDS pathogenesis, such as CD4, as well as the role played by various viral proteins and factors, for example, reverse transcriptase and protease. Little emphasis has been placed on the pathogenic aspects of the virus after chromosomal integration takes place. When one considers the role of the cytokine interleukin-1 and of glucocorticoid hormones in the pathogenesis of AIDS, it becomes increasingly apparent that the human genome, specifically the long arm of chromosome 2, where interleukin-1 is produced, may play an active role in human immunodeficiency viral, or HIV, infection, and may explain the species-specific nature of the virus. This view may lead to a paradigm shift in the understanding of AIDS pathogenesis, and produce new therapeutic tools in the treatment of HIV infection. In preview, let's take a look at the way interleukin-1 may influence HIV and the human genome. Don't worry if this may seem to be a little confusing at first. I'm going to show a figure that we will return to later following further explanation, and I think it will all fit together. So bear with me, and let's get started. It has been previously observed that HIV TAT protein binds to the long terminal repeat RNA, including the trans-acting responsive or TAR sequence, to initiate replication. The promoter region of the long terminal repeat, or LTR, is also stimulated by TAT to increase HIV replication. TAT-TAR transactivation by HIV-1 has been observed to require a factor encoded on the long arm of chromosome 2. It is now proposed that this factor may be any of several protein candidates that influence HIV and immunity, including interleukin-1, interleukin-1 receptor antagonist, which is a protein that binds to interleukin-1 receptors and inhibits the binding of interleukin-1, and or CD26, which is a lymphocyte cell surface antigen which is increased during T-cell activation. All three are produced on the long arm of chromosome 2. Now that I've confused everyone, I think what we really need to do at this point is take a look at the HIV genetic structure, and then we can go back to the figure that I had just shown you, and it would become much more understandable at that point. Let's take a look at the HIV genetic structure. To fully understand the significance of the interaction of HIV with the human genome, particularly with human chromosome 2, one must first review the genetic structure of HIV. Although a detailed examination of HIV gene structure is beyond the scope of this presentation, a general understanding of HIV structure is helpful in illustrating the methods employed by HIV to utilize human interleukin-1 and glucocorticoid hormones for its own benefit. The HIV particle, as shown in figure 1 on the top, consists of an RNA protein core surrounded by a lipid membrane containing glycoproteins, or GP, 41 and 120 that interact with CD4 antigens on the surface of immune cells. Closer examination of the RNA core, which is seen in figure 2 below that, reveals that HIV consists of 9 major genes flanked by long terminal repeats, or LTR, at either end. Closer examination of the long terminal repeat is shown in figure 3. In other words, this and this, the 2 long terminal repeats, are shown as this entire figure 3. In overview, the 9 major genes of HIV are, now we're looking at figure 2, after the long terminal repeat, or LTR, the 9 major genes are GAG, GAG, which codes for the precursor of the internal structural proteins of the virus, including P7, P9, P15, P17, and P24. The next gene is POL, P-O-L, which codes for the viral replicative proteins, protease, reverse transcriptase, integrase, and ribonuclease. VIF, which produces virion infectivity factor that increases viral infectivity. VPR, which produces viral protein R that accelerates replication. TAT, T-A-T, the transactivated gene that is essential to HIV replication and increases the rate of its own synthesis, as well as that of all viral proteins. TAT is very important to HIV. REV, R-E-V, which regulates the accumulation of unspliced or partially spliced mRNA. VPU, which produces a protein that enhances viral release from the cell. In the envelope gene that codes for the envelope proteins GP41 and GP120. And last but not least, NEF, which produces a protein that appears to suppress HIV infectivity. That is, in its absence, infectivity appears to be enhanced, thus suggesting a negative regulatory role. Now, let's move on down to figure 3, and we will discuss the major genomic regions of the HIV long-terminal repeat. And they consist of NRE, which is a negative regulatory element, very similar to NEF in many ways, because when it is deleted, one observes an increase in HIV long-terminal repeat gene expression. Interleukin-2 and interleukin-2R homology region, that's represented by IL-2 and IL-2R homology there. And this is a region that consists of two sequences that share significant homology with the interleukin-2 and interleukin-2 receptor regulatory regions. Further downstream, we see in the long-terminal repeat the enhancer region, which enhances gene expression of the promoter region. Next is the promoter region, and as you can see, it consists of 3 tandem sites that bind the cellular transcription factor SP1. In other words, this is a cellular transcription factor, SP1, and it binds to the 3 sites on the promoter region of the HIV long-terminal repeat. Site 3 has the highest affinity for SP1. Its affinity is much higher than that of site 1 or 2. Next is the TATAA, which is the region that determines proper initiation of transcription, and that's seen in other retroviruses as well. And following the RNA cap is the TAR region, or the transacting responsive region, that serves to suppress viral RNA expression and prevents RNA exit from the nucleus except in the presence of TAT protein. In other words, TAT protein binds to TAR and then the suppression of HIV viral expression is no longer in place. And remember, TAT protein binds directly to TAR RNA, which is important to know. And also, once TAT binds to TAR, RNA is at that point able to exit from the nucleus. So the TAR region is very important as well. It will be, it will become apparent that the HIV genome closely depends on the human cellular production of interleukin-1 and glucocorticoid hormones to stimulate and maintain its life cycle. This observation may lead to vulnerable targets for therapeutic intervention that are not subject to mutation and or the elusive genetic diversity of the virus. Now, let us consider the possible candidates produced on chromosome 2 that facilitate TAT-TAR transactivation. As previously mentioned, there are several products of the long arm of chromosome 2 that may play a role in HIV pathogenesis and, in fact, may be the required factor for TAT-mediated transactivation of HIV replication that is mediated via the TAR region of the long terminal repeat. Any combination of the following products may theoretically facilitate AIDS pathogenesis. If the required factor produced on the long arm of chromosome 2 for HIV TAT-mediated transactivation is interleukin-1, it would explain the observed increase in HIV replication in the presence of interleukin-1, as well as the observed increase in interleukin-1 production in the presence of HIV. Interleukin-1 may also be stimulated by HIV through the blocking of melanocyte-stimulating hormone. Now, this is a figure from the work I had published in 1996 in Medical Hypotheses. And this is to bring home the point that not only does HIV stimulate interleukin-1, but interleukin-1 may also be stimulated through a different route, which is through the blocking of MSH, or melanocyte stimulating hormone, which is actually an interleukin-1 inhibitor. So therefore you're blocking the inhibitor of interleukin-1, therefore causing an increase in interleukin-1. This occurs theoretically, but this occurs via the interaction of the peptide T subregion of GP120 with MSH receptors. Peptide T actually is very similar to alpha MSH, and it has sequences and shares has quite a bit of homology with MSH, and therefore has the capacity to interact with MSH receptors. Additionally, the induction of glucocorticoid hormones by interleukin-1 further stimulates HIV infectivity via the virion infectivity factor, or VIF, region, as we had seen, or at least had viewed when we looked at the genetic structure of HIV. Again, the figure shows the peptide T region of GP120 interacting with MSH receptors, causing a disinhibition of interleukin-1. Interleukin-1, in turn, causes an increase in corticotropin-releasing hormone, adrenocorticotropic hormone, and cortisol and or other glucocorticoid hormones. The glucocorticoid hormones, in turn, stimulate HIV in the virion infectivity factor region, and this is what plus 5002 means. It's the glucocorticoid responsive element of HIV-1, which has been identified in the VIF region of the HIV genome. So it's almost a circle, if you will. In addition, I'd like to point out at this point that both glucocorticoid hormones and interleukin-1 are able to increase CD8 positive or T suppressor lymphocytes, and they stimulate suppressor cells and cause proliferation of these types of cells, which definitely can contribute to immunosuppression. Very neat process, a very neat process that is almost self-explanatory. This is a figure that was published in my 1997 paper that was in Medical Hypothesis in May of this year, of 1997, which shows that HIV infectivity would also be stimulated if the required factor encoded on chromosome 2 were CD26. I mean, it shows other things as well. But since we are discussing whether what could be the possible factor on chromosome 2 that could cause an increase in interleukin-1 and in HIV infectivity, we should point out that chromosome 2 also produces CD26, which is also known as dipeptidyl peptidase 4. CD26 may serve as a co-receptor with CD4 that interacts with the V3 loop in GP120 of HIV and may be the key to active infection. Attachment to CD4 does not actually cause active infection, and it is believed that a cofactor is required to produce active HIV infection. Interleukin-1 may also increase HIV infectivity by stimulating CD26. So not only does chromosome 2 cause the production of CD26, but interleukin-1, which is also produced on chromosome 2, can cause an increase in CD26 production as well. Finally, and we will continue to look at this figure, if the required factor is interleukin-1 receptor antagonist, it would explain the observed homeostatic inhibition of interleukin-1 receptor antagonist by HIV, resulting in the disinhibition of interleukin-1 synthesis. This is based on the finding that antiretroviral treatment of HIV-infected subjects results in a decrease in interleukin-1 and an increase in interleukin-1 receptor antagonist. In other words, we give people who are infected an antiretroviral medication, and we notice that interleukin-1 goes down and interleukin-1 receptor goes up. thus implying that or implicating an inhibitory role of HIV in interleukin-1 receptor antagonist production. If the initiating factor for TAC-mediated transactivation is interleukin-1 receptor antagonist, HIV may regulate this by reducing interleukin-1 receptor antagonist through feedback inhibition, thus inducing interleukin-1 for the enhancement of its own replication. Interleukin-1 in turn stimulates HIV in the enhancer region of the long terminal repeat. And as a quick review of this, again, we have the lymphocyte, chromosome 2 within the nucleus, which is not pictured, of course, but this is the nucleus with chromosome 2. HIV, well, there are two HIV here. One is about to infect the cell by attaching to CD26 along with CD4, which is not shown, but the V3 loop of GP120 is interacting with CD26 to attach to and interact with the cell surface antigen to eventually cause active infection. Once the HIV is, in fact, in the cell and has begun the process of actively infecting the cell, the TAR region somehow interacts with a product of the long arm of chromosome 2. We're thinking that it could be interleukin-1 receptor antagonist that it is interacting with which it interacts, I should say. Interleukin-1 receptor antagonist, which is produced on the long arm of chromosome 2 at 2q14.2, is inhibited, I am proposing, by HIV. This inhibition causes a reduction in the amount of interleukin-1 receptor antagonist. Therefore, there is going to be a disinhibition of interleukin-1. You're going to have more interleukin-1 produced, which is what we see when the opposite is true, that when a person is given an antiretroviral medication, interleukin-1 is reduced and interleukin-1 receptor antagonist is increased. We're saying that when HIV comes in, interleukin-1 receptor antagonist is reduced and interleukin-1 is increased. The interleukin-1 excess not only causes HIV to be stimulated in the enhancer region, but also it causes an increase in CD26, thus increasing HIV infectivity via the V3 loop region of GP120 of the virus. So not only is replication enhanced, but infectivity is enhanced as well. Have led me to believe that interleukin-1 receptor antagonist is a likely factor involved in HIV infection and in AIDS pathogenesis. As previously mentioned, HIV apparently inhibits the production of interleukin-1 receptor antagonist. Interleukin-1 receptor antagonist is produced on the long arm of chromosome 2 at 2q14.2. It is now proposed that interleukin-1 receptor antagonist is the major factor encoded on the long arm of chromosome 2 that is required for TAR transactivation and is controlled by HIV through feedback inhibition. Again, this is supported by the observation that antiretroviral treatment in HIV-infected individuals results in the disinhibition of interleukin-1 receptor antagonist. thus implying an inhibitory role of HIV in the production of interleukin-1 receptor antagonists. The inhibition of interleukin-1 receptor antagonists by HIV would give greater control to the virus in determining its rate of replication via the disinhibition of interleukin-1, thus initiating a cascade of observed benefits to HIV. Let's take another look at Figure 4 once again. Figure 4, which I had shown at the beginning of this tape, will now seem to make much more sense. At least I hope it does. When we take another look at this, we can see that HIV uses interleukin-1 for its own benefit. And some of these benefits include the stimulation of HIV replication via the long terminal repeat enhancer region by interleukin-1. In other words, interleukin-1 is stimulated, and interleukin-1 stimulates HIV in the enhancer region of the long terminal repeat. Also, the induction of glucocorticoid hormone synthesis by interleukin-1 that stimulates HIV in the virion infectivity factor region, as indicated here. Increased viral infectivity via stimulation of CD26 by interleukin-1, which is not shown in this particular figure. There's also a gradual reduction of CD4 lymphocytes and rapid expansion of CD8 lymphocytes as well, as we have seen in the one of the previous figures. This would also explain the resistance of glucocorticoid resistant cells to HIV infection, as seen by Kawa et al. 1993, in which the virus is unable to or unlikely to infect a cell that is resistant to the very glucocorticoids it requires for replication and survival. Additionally, HIV viral protein R, or VPR, which accelerates viral replication and activates HIV-1 LTR, or long terminal repeat, in a very similar way as the TAC protein that does. In fact, also interacts with glucocorticoid receptors type 2, as seen by Raffaelli in 1995 et al. Raffaelli et al., I should say. And VPR is the only regulatory HIV-1 protein to be actually incorporated within the virus. Considering the importance of glucocorticoid hormones in the pathogenesis of HIV infection and AIDS, it is likely that VPR is directly involved in the establishment of infection as well, and plays a role in the observed selection of glucocorticoid sensitive cells by HIV. Now, it seems that Figure 4 should make a lot of sense. Since we know the HIV viral genome, we know the long terminal repeat genome, we know that in the lymphocyte, human chromosome 2 is actively involved in the process of TAT-mediated transactivation. This is the TAT protein. It's going to the long terminal repeat. It stimulates both the promoter region and the TAR, or the transactivation responsive region. TAR interacts with something. We don't know what it is, but we believe it's interleukin-1 receptor antagonist that comes from human chromosome 2. That interaction in turn results in the disinhibition of interleukin-1. Interleukin-1 in turn causes a stimulation in corticotropin releasing hormone and adrenocorticotropic hormone, resulting in an increase in glucocorticoid hormones. Interleukin-1 stimulates the enhancer region of the long-terminal repeat. The glucocorticoids stimulate the virion infectivity factor region. It's a circle. It's a very, very clear circle that HIV utilizes human interleukin-1, glucocorticoid hormones, and human chromosome 2 to perpetuate its own existence. If the caption is not clearly seen on the tape, I will read that for you as well. Basically, I had written that HIV TAC protein binds to the promoter and TAR regions of the LTR. TAR interacts directly with a factor encoded on the long arm of chromosome 2, possibly interleukin-1 receptor antagonist, to increase interleukin-1 and glucocorticoid hormone synthesis, which in turn stimulate HIV replication and infectivity via the enhancer and VIF regions, respectively. In conclusion, when one views the process of HIV infection as being mediated by interleukin-1 and glucocorticoid hormones, one has a reasonable explanation of the entire disease process, including the relentless rate of viral infectivity, the expansion of CD8 cells, the depletion of CD4 positive cells with the resultant immunodeficiency, and the observable therapeutic use of ascorbic acid, which has been found to inhibit the release of corticotropin-releasing hormone, as well as inhibiting HIV replication. My guess is that because ascorbic acid or vitamin C can inhibit corticotropin-releasing hormone, it therefore is able to suppress HIV replication. Just as rhinovirus has an affinity for respiratory mucosa and hepatitis virus has an affinity for hepatic cells, HIV apparently has an affinity for lymphoid cells and targets the genetic machinery associated with human interleukin-1 and glucocorticoid hormones to facilitate its replication. The development of the clinical features of AIDS is merely a consequence of this process, an unfortunate result of HIV's affinity for and ability to use human interleukin-1 and glucocorticoid hormones for reproduction. Significantly, the host may succumb to this process long after the progeny of HIV have been passed on to subsequent hosts. HIV is evidently successful for this reason. Only when new HIV infections are prevented and treatment of those already infected addresses the HIV-induced interleukin-1 and glucocorticoid hormone excess will a permanent and substantial dent be made in the defenses of this most difficult viral adversary. If there are any further questions that you may have or comments, please feel free to write to me at the address seen at the beginning of this tape, which is 110-64 Queens Boulevard, number 129, Forest Hills, New York, 11375, USA. Thank you very much, and it is my hope that together we can resolve this problem in a way that can be of benefit to all. Thank you.